Hello guys, welcome to Science Learning Gateway. So I hope you all are doing good. So today in this tutorial we are going to discuss class 10 CBSE term 2 for class 10 CBSE term 2 syllabus, and we are going to discuss chapter number 9, heredity and evolution. As you know that in the latest syllabus you have to read only heredity part. So in this chapter we are going to read only heredity part, and in one shot we'll finish this chapter completely. And some application based questions can come from this lesson, and maybe 1 marks or 2 marks questions can come. So be ready with the chapter, go through the chapter properly, read your NCERT textbook, because from your NCERT textbook only you're going to get the questions in the exams. So let's get started. So first of all, we should know the definition of genetics. The first topic which is given in your textbook is genetics. So what is genetics? It is a branch of science that deals with the study of heredity and evolution. As we have different branches of science like physics, chemistry, biology, like that. In the same way, we have one branch of science called genetics, in which we have to read about heredity and evolution. So it is a branch of science which deals with the study of heredity and variation. Okay, heredity and variation. So now let's discuss what is heredity and what is variation. So what is heredity? The transmission of characters, or you can say transfer. You can say transfer also. The transmission or transfer of characters or traits from one generation to the next generation is called heredity. For example, this is your parents' generation and this is your generation B. So some characters are passed from your parents to you. For example, your eyes color are similar to your parents, or your height is similar to your parents. So that character or trait has been passed on from your parents to you people, right? So heredity is the transfer of characters from one generation to the next generation. Next we have variation. So variation means what? Variation means differences, correct? So what is variation? The differences in the characters or traits between the parents and the offspring is called variation. For example, we are not exactly similar to our parents. Some characters are different between us and our parents, right? So that differences in characters or traits between the parents and the offspring is called variations. And there are two types of variations: somatic variations and gammatic variations. So let's discuss what is somatic variation and what is gammatic variation. The direct points I have given you over there. If questions will be asked what is heredity, define variation, you can write exactly the same answer in your answer sheets. Now what is somatic variation? It occurs in somatic cells, right? The word somatic is given over there, so it will occur in somatic cells. They are not transmitted in the next generation. They are also known as acquired traits. Now when I have explained once again this complete chapter, that time I have told you, there are two types of traits: acquired traits and inherited traits. So inherited traits are those traits which we get from our parents, for example eye color, our height, skin color, etc. But acquired traits are those traits which we develop in our lifetime, for example our learning power, development of muscles, etc. Right? So somatic variation occurs in somatic cells and they are not transmitted in the next generation, means this we will not get from our parents, and they are also known as acquired traits. Now let's discuss about gammatic variation. So gammatic variations occur in germ cells, or you can say reproductive cells. They are transmitted or inherited in the next generation, means this we can get from our parents, right? These variations means these differences are passed on from one generation to the other generation, and they are also known as inherited traits. So remember that somatic variations are known as acquired traits, gammatic variations are known as inherited traits. Now let's discuss about genes. So as you know that genes are the functional unit of heredity, right? Genes are passed on from one generation to the other generation, and it also controls the particular characteristics in an organism: eye color, height of the person, skin color, etc. These all characters which we have in our body are controlled by the genes, because they are passed on from one generation to the other generation. So remember that genes are the functional unit of heredity, and it controls the characteristics in living organism. Now let's go to the next slide, in which we will read about Mendel and his work on inheritance. So over there, Gregor John Mendel in the year 1822 and 1884 started his experiment on a plant breeding and hybridization, and he is also known as Gregor John Mendel. He is also known as father of genetics. From here, 1 marks question you can get that who is known as father of genetics. Maybe chances are there. So remember that Gregor John Mendel is known as father of genetics. Right now, some laws he has given. He has proposed a law of inheritance in an organism. So from the word inheritance, what you can understand, inheritance means what we get from our parents, right? So according to law of inheritance, the characters are passed on from one generation to the next generation. Now there are three basic laws of inheritance: law of dominance, law of segregation, and the law of independent assortment. So let's start with law of dominance. So what is law of dominance? According to law of dominance, some alleles are dominant and cover up the recessive alleles. There are two types of traits, dominant and recessive one. So dominant traits will always be stronger than the recessive traits, right? So according to law of dominance, dominant one means the stronger one. So some alleles are dominant and cover up the recessive traits, means the dominant trait will show its character in the next generation. Now let's come to the law of segregation. Segregation means it will break or separate. So according to law of segregation, an organism has two alleles for each gene, but they can only pass on one, right? So two alleles are there. For example, T and T. So only one allele will pass on to the next generation. Next, according to law of independent assortment, what we have read, that genes are found on separate chromosomes are inherited independent of each other. Now, you know that genes are present in the chromosomes, right? And they are passed on from the parent to their offspring. Independent assortment means they will not depend on each other. They will independently passed on to the next generation. Now this Gregor John Mendel, he has selected which plant for his experiment? He has selected pea plant for his experiment. And what is the scientific name of pea plant? Pisum sativum. Pisum sativum. So remember that which plant Gregor Mendel has selected for his experiment? He has selected pea plant. And the scientific name of pea plant is Pisum sativum. So you have to remember that, this one. Now, and why he has used this pea plant for his work? Because there are some characters which are present in this pea plant which help it to carry out its experiment. First character is that this pea plant has 7 pairs of contrasting characters. How many pairs? 7 pairs of contrasting characters are present in pea plant. For example, flower color, flower position, seed color, seed shape, pod color, and height of the plant. You can see in this table that all the 7 pairs of contrasting characters are present over there. So this was the first point, at why he has selected pea plant. The second point was that pea plant can grow very easily. It can grow very easily. That was the second point. And the third point is that it has a very short life cycle, right? It has a very short life cycle. So these are the 3 points why Gregor Mendel has selected Pisum sativum for his experiment. Now if we talk about the flower color, then there are 2 characters. Violet color is a dominant trait, and for recessive trait, white color. 2 types of flower positions are there, axial and terminal. Then seed color is also yellow and green. Yellow is a dominant one and green is the recessive one. Then if we talk about the seed shape, round seed and wrinkle seeds are there. Pod shaped, inflated. Inflated means like this, and constricted means this type of peas we can see in the market. Next is pod color. It can be green or it can be yellow, and height of the plant can be tall and can be dwarf, short also. So these are the 7 pairs of contrasting characters. As you all know that contrasting characters are also present in humans. For example, suppose our father has brown eye color and our mother has black eye color, right? So those are the contrasting characters. So in the same way, in pea plant also 7 pairs of contrasting characters are present. Now, see, the points are also given over there, that why Gregor Mendel has selected pea plant for his experiment, right? So this question, maybe chances are there that it can also come. We can't say anything. So why Mendel has selected pea plant for his experiment? The first point is that they are easy to grow and have very short lifespan, right? This is the first point. Second point, the flowers of these plants are bisexual. Bisexual means both male and female characters are present in the flowers of pea plant. Second, Third point, they produce large number of seeds. Yes, we know that in pea plant lots of seeds are there. In one pea plant we can get so many pea seeds, right? Both self and cross pollination is possible in pea plant. And the last point is different characters present in pea plant can be easily recognized. Means 7 pairs of contrasting characters are there, so that we can easily recognize in a pea plant. Next, now let's study about the Mendel's experiment. So what he did, he has conducted a series of experiment in which he has crossed the pollinated plants to study one character at a time. So first of all, we'll read about monohybrid cross, and after that we'll read about dihybrid cross. So in monohybrid cross, what he did, in monohybrid cross, Mendel has taken two pea plant with one pair of contrasting character. For example, they have taken the height of a plant. One tall plant they have taken and one dwarf plant they have taken. So let's monohybrid means only one pair of contrasting characters. So see, you can see over there one tall plant is there, and we can write we can represent a tall plant with capital T, capital T. And the dwarf plant can be represented as small t, small t. The alleles, the traits which are responsible for the tall characters are capital T, and for the short or the dwarf plant, small t and small t. So if the cross will be there between tall and the short plant, in F1 generation, what we will get? All the plants will be tall. Okay, how we can get that? Suppose this side I have taken this one, and this side I have taken capital T, capital T. So once this T, and again if this will come capital T, small t, and yeah, again you can cross 1 2, again you can take a cross between this tall plant and this dwarf plant, and this and this. So all the plants which you can see over there, they are tall because the tall characters are present in all the 4 plants. So in F1 generation, the plants which you will get will always be tall. Now let's come to the next one. In F2 generation, what are the changes which we can see? So now let's do a cross between a tall plant and another tall plant which we have got in F1 generation. So the traits are capital T, small t, and again capital T and small t. So let's have a cross. First this T and this T will cross together, so we will get a tall plant. Then again this T and this T will cross, then capital T, small t, this small t and capital T, then again capital T and small t, and after that small t, small t. So 3 tall plants we have got in F2 generation, and only 1 dwarf plant we have got in the F2 generation. So our ratio will be 3:1. This is the phenotypic ratio which we will get in the monohybrid cross. So I hope you have understood that in your monohybrid cross, in F1 generation all the plants will be tall, but in F2 generation 3 plants will be tall and 1 plant will be dwarf one. You can see over there. So see, in a monohybrid cross, the cross was between a tall plant and a dwarf plant. So tall plant is represented as capital T, and a dwarf plant is represented as small t, small t. So one characters will come from everywhere. From here we will take capital T, from here we will take a small t, right? So when it will cross, we will get this character T and T. So means all the plants will be tall in F1 generation. But in F2 generation, what will happen when the cross will be carried out? 3 plants will be tall, but 1 plant will be a dwarf plant. So we can write the genotypic ratio as capital T, this T and this T. That is one tall plant we have got, 1 completely tall plant we have got, 2 with mixed character and 1 with small character. So 1:2:1, and the tall is to dwarf character. How many tall plants we have got? We have got 3 tall plants and 1 dwarf plant. So phenotypic ratio will be 3:1 and the genotypic ratio will be 1:2:1. Phenotypic means the physical characters, what we can see. We can see that over there the 3 tall plants are there. So our phenotypic characters will be 3:1, but the genotypic character will be 1:2. See, this only 1 trait is there and these 2 are similar. So that's why we have written 2 and over there only 1. So 1:2:1 is our genotypic ratio. So that's all about your monohybrid cross. So remember that in monohybrid cross, in F1 generation, all the plants will be tall, but in F2 generation, there will be 3 tall plants and 1 dwarf plant. Now let's read about the dihybrid cross. So in dihybrid cross, we have to take 2 pairs of contrasting characters. Right? So a cross between 2 plants having 2 pairs of contrasting character is called a dihybrid cross. So let's take a seed which is green and round in shape, which is round and yellow in color, and other one is round and let's take a seed which is round and green in color, and the another seed is wrinkled and yellow in color. So round is a dominant trait and yellow color is a recessive trait. Again green color, round shape is the dominant one and wrinkled is the recessive one. Green color is the dominant one and yellow color is the recessive one, right? So this is the seed characters. So this is the parental generation, right, in which the color of the seed is green and it is round in shape, and in this the color of the seed is yellow and it is wrinkled in shape. So the gametes which will come from these two are only one capital R, and from these two y, small y will come. From here one character will come, one small r and one capital Y. So in F1 generation, if we do a cross between them, we will get a seed which is round and yellow in color, right, which is round and yellow in color. Capital R, small r. From here the capital R is the dominant one, so the shape of the seed will be round, and capital Y and small y, yellow color is the dominant one, so the color of the seed will be yellow. Now in F1 generation, what we can see that only 2 seeds all the seeds which were developed in F1 generations are round and yellow in color. Now we have to do a cross between these two plants in F2 generation. That is called a Punnett square. So you can see a Punnett square is made over there. Over there we will take the characters. One capital R and one capital Y we can take. One capital R and one again one small y we can take, right, and small r, capital Y, small r and small y. So this side also will write the same thing. If a cross bit will be done between capital R, you can get RRYY. Again this cross and this cross will be done, you will get this thing. This will cross with this, you will get this one, and this will cross with this, you will get this character. So like this you have to do for this, and like this you will complete this Punnett square, right? So a detailed video I have already uploaded this in the playlist of class 10. You can go and check that, and you can see over there in this all the seeds are round and yellow in color. Over here also the seeds are round and yellow in color. Over here also the seeds are round and yellow in color, but over here also the seeds are round and yellow in color. Over here also the seeds are round and yellow in color. This is not round and yellow. This is round and yellow in color. Round and yellow in color. 1 2 3 4 5 6 7 8 9. 1 2 3 4 5 6 7 8 9. Yeah, so there are 9 seeds which are round and yellow in color. Means round and yellow are the dominant trait which we can see in the F2 generation. Next, there are 3 seeds which are round and green in color, and there are 3 seeds which are wrinkled and yellow in color, and there is only one seed that is wrinkled and green in color. You can see small r. This is for your green color, and small y is for yellow one. So this is the seed which one is wrinkled and green in color. This is a recessive trait showing only one seed over there. So your phenotypic ratio will be 9:3:3:1 in this case, in the case of dihybrid cross. Now, how do these traits get expressed? Let's read it once. The cellular DNA is the information source for making proteins in the cell. Yes, you know that DNA is most important, which plays a very important role in passing the characters from one generation to the next generation. So it makes the protein in the cell. A section of DNA that provides information for one protein is called the gene for that protein. And inside the DNA, what is present? Gene is present. For example, tallness. as a characteristic in plant. You know that tallness is one of the characteristic in plant. Next, plants have hormones that can trigger the growth. Plant height can thus depend on the amount of particular plant hormone. The amount of plant hormone made will depend on the efficiency of the process for making it. Next, the hormones which are present in plant that also play a very important role in the growth of the plant. For example, auxins. It is one of the growth hormone. An enzyme is important for this process. You know that enzymes play a very important role in carry out the reaction, right? Enzymes work is also important over there. But enzyme will work efficiently, a lot of hormone will be made and the plant will be tall. If the enzyme will work in a very efficient way, what will happen? Hormone will be made in a good quantity and the plant will be tall. But if the enzyme will not work properly, the amount of hormone secreted will be less and the plant will be short in height. Thus, genes control characteristics or traits, right? So genes play a very important role in controlling the characteristics or traits in a particular plant or any organism. Now the last topic from here is sex determination, and this is one of the most important topic from this chapter, heredity and evolution. Many times 5 marks questions are asked from this topic, so listen to it very carefully. So sex determination. Sex determination means whether the child is a girl child or a boy child. That is the meaning of sex determination, correct? So sex determination is a process in which sex of offspring is determined. Offspring means the baby, correct? Sex means whether the child is a girl child or a boy child. That is, sex determination is that process in which we check whether the child is a girl child or a boy child. So there are two factors which are responsible for sex determination. So what are the two factors? The first factor is environmental factor and the next factor is genetic factor. So what is environmental factor? In some animals like reptiles, temperature at which fertilized eggs are kept determines whether the offspring will be a male or female. So, see, in some of the organisms, for example, if we take the example of reptiles, the temperature at which the female reptile will the temperature at which the eggs get fertilized, at which the fertilized eggs are kept, determines whether the offspring will be a male. Means temperature plays an important role over there. So to determine that the reptile which will be produced will be a male or a female. So environment is playing an important role over there, but genetic factor is the most important factor, which we can see in human beings, right? Because in human beings we have sex chromosomes, right? How many chromosomes are there in human beings? We have 46 chromosomes. That is, we have 23 pairs of chromosome. We have 23 pairs of chromosome. Out of 23 pairs, 22 pairs are called autosomes. 22 pairs are called autosomes. And one pair of chromosome is called sex chromosome. So this sex chromosome plays a very important role in sex determination of a child. So in male, the one pair of chromosome which is present is X and Y. This is the one pair of chromosome which is present in male. This is a sex chromosome in male. But in female, the sex chromosome which is present is in the form of X and X. XX. Similar types of chromosomes are present in male, female, but in male the chromosomes are different. That is X and Y. So now let's read about the sex chromosomes. So in reptiles, environmental factors play an important role, that the given offspring will be a male or a female. But in humans, genetic factor plays an important role, right? Now let's read about sex chromosomes. So in human beings, how many pairs of chromosomes are there? 23 pairs. Remember that. Out of 23 pairs, 22 pairs are called autosome, and one pair of chromosome which determines the sex of the given offspring is called sex chromosome. So in female, which type of sex chromosomes are there? XX, and in male the chromosome is X and Y. So women have a perfect pair of sex chromosome, both called X, but men have a mismatch pair in which one is normal size X and the other is a short one Y. So sometimes the question is asked that what is the difference between the sex chromosome of male and female. So you can write that in female the sex chromosomes are similar. Sex chromosomes have a perfect pair called XX, but in male the sex chromosomes are different. One is of normal size and one is of short size, that is X and Y. So this is the difference between the sex chromosome of male and female. Now let's read that how the sex of a child is determined. Sex determination in humans. This is the last topic which you have to read for your term 2 syllabus. See, you can see in this figure, the male is carrying which type of chromosome? X and Y sex chromosome, but the female is carrying XX sex chromosome. Now, the gametes, when the gametes will be formed in a male, some of the gamete will carry X chromosome and some of the gamete will carry Y chromosome, right? Lots of gametes are formed in male, so some of the gametes will get X chromosome and some of the gamete will get Y chromosome. But in female, only one egg is formed, and since they have XX chromosome, same type of chromosome, the egg will carry only X chromosome along with it. So if the gamete carrying X chromosome will will fertilize with the egg carrying X chromosome, the zygote formed will be XX. The zygote form will be XX. But if the gamete carrying Y chromosome will fertilize with the egg carrying X chromosome, the gamete form will be X and Y. If the zygote will get XX chromosome, the offspring form will be a female child, because in female similar pair of sex chromosomes are present, that is X and X. But the zygote, if it will get XY chromosome, then the male child will be formed. So once again we will do this one once again. This is for male and this is for female. Males carry a pair of XY chromosomes and female carries a pair of XX chromosomes. So the gametes will carry X chromosome and Y chromosome, but from female similar chromosomes are there, so they will carry XX. If X chromosome of male will fertilize with the X chromosome of female, XX type of zygote or the offspring will be formed. So that will be a female child. But if Y gamete will cross with the X gamete of female, then the child will get XY type of combination. So this will be for a male child. So who is responsible for the characters, or means who is responsible over there, that what type of sex will be present in the given offspring? Obviously the father is responsible for that, because the father is having different types of sex chromosome, that is X and Y, but the mother is having the similar types of chromosome. So father is responsible for the determination of sex in a child, right? But in olden days what was happened, that only females are responsible, that you have given birth to a girl child. But it's not true. The father is responsible for the determination of sex in a child, whether it is a boy child or a girl child, right? So that's all which we have read today. So in 26 or 27 minutes we have completed the full chapter, correct? So I hope you all have understood this. So let's give a short recap what we have read in this lesson. So first of all, we have read about genetics. It is a branch of science which deals with the study of heredity and variation. Next, we have read about heredity. What is heredity? The transfer of characters from one generation to the next generation is called heredity. And variation means the differences which are present between the parent and the offspring. There are two types of variation: somatic variations and gammatic variations. Somatic variations occurs in somatic cells. Gammatic variations occurs in germ cells. Now, somatic variations are not passed on to the next generation. They are also called acquired traits, and gammatic variations are passed on from one generation to the other generation. They are called inherited traits. If we talk about traits or characters, then our eye color, height, skin color, they all are called traits or characters, and there are two types of traits: acquired traits and inherited traits. Acquired traits are those traits which we develop in our lifetime. For example, learning power, development of muscles. And your inherited traits are those traits which we get from our parents, for example eye color, skin color, etc. Next, genes are the functional unit of heredity which controls the characteristics in a living organism. Next, we have read Mendel and his work on inheritance. So Mendel is also called father of genetics. He has selected pea plant for carrying out his experiment, and the scientific name of pea plant is Pisum sativum. So because of some characters, Mendel has selected garden pea plant. Why? Because the pea plants can grow easily, the flowers are bisexual, they produce large numbers of seeds, and the most important is that pea plants are having 7 pairs of contrasting characters. In monohybrid cross, what we have read in monohybrid cross, there are the cross between two pea plants having one pair of contrasting characters is done. In F1 generation, all the plants were tall, but in F2 generation, 3 tall and 1 dwarf plant can be seen. But in dihybrid cross, there are again the cross was done between two plants, but having 2 pairs of contrasting characters. But in dihybrid cross, what we have seen in F1 generation, all the seeds were of brown and yellow in color, but in F2 generation, the ratio was 9:3:1. Next, we have read about sex determination, that in there are two factors which are responsible for sex determination: environmental factors and genetic factors. So over there, sex determination in human is most important. That in human, 23 pairs of chromosomes are there. Out of 23 pairs, 22 pairs are your sex chromosome, and 22 pairs are called autosomes, and one pair is called sex chromosome. Next, what is the difference between the sex chromosomes of male and female? Males are having XY type of combination, they are having XY sex chromosome, but females are having XX types of combination which are similar. Now, who determines the sex in a child? Father is responsible for determination of sex in a child, because father carries a different type of chromosome X and Y, but mother carries a similar type of chromosome, that is X and X. Right? So that was all about this chapter, heredity. I hope this was clear to you. If you have any doubt, you can comment in the comment section. I will definitely give a reply for your queries. So thank you for watching. If you are new to my channel, then please subscribe to it, and don't forget to hit the bell icon so that whenever I will be uploading a new video, you will get the notification for that. And if you like this tutorial, then please click on the like button and share it with your friends also. So thank you for watching.